Hi students, welcome to HSC Biology and Module 5 on Heredity. This has been a bit of a while between videos and I haven't been that well, so no COVID problems, but just a little bit of a cold, and so it wasn't really ideal for recording. But we're back and hopefully we'll be able to push through some of these videos in this first of the HSC modules, Module 5 Heredity. This is number 13. Video 13, and we're going to be looking at contrasting DNA in prokaryotes and eukaryotes. Now what we have to do here is we have to construct appropriate representations to model and compare the forms in which DNA exists in eukaryotes and prokaryotes. And this is going to be very important for a lot of our future studies in biology as we start to understand how we've been able to manipulate genes in the genetic code in order for us to do a whole range of different things. And in order to do that, we need to actually understand what it is that we're working with. And so therefore, important to get this understanding of the difference between DNA in prokaryotic and eukaryotic organisms, or at least in different cells. So again, 3 levels that you want to try and aim for. It's important that you can at least be able to distinguish between DNA in the 2 different groups. If you can draw comparison tables, that allows you to look at similarities and differences between the DNA of each of these 2 important groups. And then obviously we want to critically evaluate models. So modeling is something that you're going to be doing a lot of in biology because we deal with so many very tiny things that we want to try and understand what's actually going on at the molecular level. And of course, one of the important things that we're aware of is that the actual structure of DNA itself was worked out through very large models designed to understand how the chemicals were interacting with one another. So let's have a look at prokaryotes and eukaryotes, and hopefully you remember these 2 terms from your Year 11 course. The important thing is that all living things have genetic molecules, information carriers, and we now know that is the DNA. This is a fantastic molecule, not only because it's capable of replicating itself, and copies are so very important. Being able to maintain the integrity of that is important during a copying process, but also the fact that there has to be information. There needs to be information carried in that molecule that actually helps us to explain why bacteria, for example, a prokaryotic organism, looks so different to us. And that has to be because there must be some information that's being carried in the cells of the bacterium that are very different to the information that's being carried in our cells, and yet are based on similar sort of a structure. And that's what DNA does for us. DNA allows us to have a huge amount of variation within a very common structure, and it's that 4 different types of nitrogenous bases and the way they combine together that helps us explain not only replication but also why that information can differ from one organism to another. So when we look at the composition of DNA, then we're looking at some quite basic similarities between the chemical composition or the chemical nature of DNA, particularly in terms of a nucleotide. We know that the nucleotides are these kind of base units of DNA, which is phosphate connected to a sugar, connected to a base. And the bases we now know are G's, C's, A's, and T's, and they bond in that way. But of course, the order of the base pairs is what is different. And so when we look at a sequence of DNA, and of course we tend, when we're discussing DNA, to discuss one strand. We know that it's a double strand, it's a double helix, 2 strands coiled around one another, and that coiling is where the base pairing occurs. But we know that the actual sequence is what differs. So, a sequence might be AAAG, AAAC. That would be one sequence. You can make any sequence that you like up based on those 4 letters in any combinations, and they are, the order of those is very, very important. However, when we look at prokaryotic cells and eukaryotic cells, we do see differences in the arrangement or the organization of the DNA. We know that in a eukaryotic cell, they are cells that have membrane-bound organelles, which means we have a nucleus, and that's where we find the DNA in the eukaryotic cell. Prokaryotic cells don't have membrane-bound organelles, so there's no nucleus, there's no particular structure, and often we find coiled DNA, simple coil DNA in something called a plasmid. And these are things that we're going to be looking at in a little bit more detail in this particular video. So let's look at prokaryotic cells first. This is a prokaryotic cell, and you can see in this particular cell the organization is quite simple. We do, or we can have cilia or beating hairs on the outside of the cell. We may also have a tail or flagellum, which may be part of our prokaryotic cell, but you can see the organization of chromosomes is very simple inside of the prokaryotic cell. So we still have DNA, and that DNA tends to be in a kind of a single loop. Sometimes it's called a nucleoid. Try and avoid using words that kind of are reminiscent of a eukaryotic organization. But there's kind of, if you think about the fact that the chromosome is very simple, it's kind of looped together inside, and there can be these smaller circular pieces of DNA called plasmids, which I talked about on the previous slide. And that's pretty much the organization of genetic material inside of the prokaryotic cell. So we find irregularly shaped regions that can be slightly denser than the rest of the cytoplasm. We find these large circular plasmids, which are the little rings of DNA, and those are going to be particularly important later on as we start to look at ways of manipulating the genetic information. And because of their rapid rate of reproduction, as well as the simplicity of their DNA, bacteria have been chosen as the subjects of a lot of very important genetic studies. Now, in eukaryotic cells, we have these histone proteins, these kind of cotton reels that the DNA is wound around. We don't have that same level of organization in the prokaryotic cells. So it's a much simpler system. It's a system that's much easier for us, as I said, to manipulate. And when you're constructing models, it's also something that you can construct on a much simpler basis than what we see when we look at the eukaryotic cell. When we look at the eukaryotic cells, of course, the key here is that we do have membrane-bound organelles, and because of that, there's an additional difference that we find that's critically important when we're studying eukaryotic cells. So we've got this nuclear DNA, and the nuclear DNA is the material that's basically been contributed by both parents. So a sperm fertilizing an egg creates a united zygote, so genetic material that has been sourced from both parents, mother and father, and that material is obviously going to replicate, and hopefully as much as possible is going to be consistent in that replication from generation to generation of the cells. But that material has been sourced from both parents. However, inside of the fertilized egg, there is also mitochondrial DNA. So DNA which has actually come from organelles within the female's cells. So nothing that's contributed by the sperm. The sperm really just contributes to that nuclear DNA and not much else. The rest of the cell is pretty much all from the mother. And as a result of that, any DNA that may be present that's not part of that nuclear DNA will be hers and hers alone, and actually can be tracked back through the maternal line. So mother to mother to mother to mother, basically. So when we're looking at mitochondrial DNA, we're actually looking at the sequence of female inheritance or female ancestry going back. And that's the way we can track through the genetic information that's being preserved from mother to daughter through the generations. Now, mitochondrial DNA isn't huge. It only holds a small number of genes, and the circular arrangement of this DNA is reminiscent of plasmids, which may also tell us a little bit of something about the origin of these particular organelles. The function of the DNA here, as you might expect with the mitochondria having such an important role to play in respiration, is coding for some of the processes that are a very important part of that chemical reaction in cells. but also involved in the building of RNA molecules. Now, we haven't looked too much at this stage at things like how we model each of these. We've really looked at just two-dimensional models at this stage. But I'm hoping that what you can do is to start to have a look at the models that you construct in class, these two-dimensional models, these drawings that we represent of these very tiny structures, and to start to get a little bit of a sense of both the utility of models in terms of how they help us explain similarities and differences between, say, the DNA of prokaryotes and eukaryotes, but also some of the limitations that we also see with models. So modeling will be something that we'll do a little more of in class. Thanks for watching.